H. Recovery of cells containing recombinant plasmid. Colonies complementary to nitrocellulose signal are recovered from the master plate and cultured in nutrient broth in order to get the sufficient bacterial clones containing recombinant plasmid. Figure 6.9g. However, in some instances the foreign DNA is cleaved enzymatically from the vector molecules. Sometimes aim lies to achieve expression of gene within the bacterial cell if it encodes for valuable polypeptides. L. Expression of cloned DNA. Factors taken into account for expression are 1. Supply of prokaryotic promoter for expression of eukaryotic genes, 2. Supply of ribosomal binding sites for the cloning vector, 3. Removal of introns from mRNA obtained from eukaryotic genes, and 4. Inhibition of gene responsible for degradation of foreign protein within the bacterium. The genes of eukaryotes differ in different ways from those of E. coli. Therefore, the eukaryotic genes must be modified in such a way that they could resemble the prokaryotic genes and express in E. coli. To begin with transcription in prokaryotes, the binding of bacterial RNA polymerase to a promoter region of the DNA immediately before or upstream of the gene is essential. 1. Shine-Dalgarno sequence. Prokaryotic mRNA have a sequence upstream from the initiation codon that play a role in attachment of the 30S ribosomal subunit to the mRNA. This sequence of about 3 to 9 bases long is about bases upstream from the initiation codon, which is also complementary to a sequence near the 3' terminus of 16S rRNA. The name of this sequence was coined as the Shine-Dalgarno sequence, after the name of the discoverer J. Shine and L. Dalgarno in 1975. For the expression of eukaryotic gene within E. coli, presence of a Shine-Dalgarno sequence in bacteria is necessary. 2. Expression vectors. In cDNA, translation is correctly started at the universal initiation codon, August. To overcome the problem of time consumption in adding a Shine-Dalgarno promoter sequence and initiation codon to every foreign gene, the expression vector has been built up. It contains a strong promoter, the activities of which are controlled by temperature, concentration of specific inducer or repressor, and a Shine-Dalgarno sequence. In addition to promoter, the expression vectors also have restriction sites, origin of replication, and marker genes for resistance against the specific antibiotics. The eukaryotic promoter is necessarily replaced with a prokaryotic promoter in order to carry out transcription of an eukaryotic gene. Hence, for the construction of expression vectors, a number of bacterial promoters have been used, for example, promoter or Lac or Trp Operon from E. coli and Sus lactamus promoter from PBr322 plasmid. The Tac promoter is a hybrid promoter constructed between elements of the Trp and Lac promoters. These promoters are regulated by their corresponding repressors. The Lac Operon is negatively regulated by the Lac repressor and can be depressed by addition of an inducer, i.e. isopropyl sus-D-thiogalactosidase (IPTG). It is positively regulated by the catabolic activator protein Chap complexed with cyclic AMP pathway. Similarly, the Trp Operon encodes 5 enzymes for the biosynthesis of tryptophan. The complex formed between the Trp repressor and tryptophan negatively control the Trp Operon. When the amino acid is in abundant supply, the genes responsible for its biosynthesis are repressed and do not work. But the eukaryotic proteins are not always stable in prokaryotic cells. It is disintegrated by the bacterium. Protection of protein is necessary. When a foreign gene is inserted in a prokaryotic gene, a hybrid polypeptide is produced. It contains a part of the prokaryotic product attached to N-terminus of the foreign polypeptide. The extra sequence of N-terminus may stabilize the polypeptide and may also interfere in the right functioning of the foreign protein. Therefore, it is necessary to separate the hybrid protein by chemical cleavage at methionine. J. Detection of nucleic acids. There are two approaches for labeling the nucleic acids to be used as DNA probe: radioactive labeling and non-radioactive labeling. 1. Radioactive labeling. Methods for in vitro introduction of radioactivity in nucleic acids was developed during 1970s through metabolic labeling. In this method, large amount of radioactivity was required. However, this method was time-consuming and laborious. The methods developed later on utilized FAGT4 polynucleotide kinase, which transfers YP04 of ATP to 5' terminus in DNA or RNA, or E. coli DNA polymerase I, which replaces a normal nucleotide of double-stranded DNA with a -32Po labeled nucleotides. The nucleic acids are labeled in vitro by using the following methods. A. Nic Translation. This method is used to label the double-stranded DNA probes. If there is no Nic, a Nic is made on double-stranded DNA by using DNase in a buffered solution containing DNTPs which exposes 3O group on one strand of DNA. All the 4 nucleotides are radiolabeled A32P DNTPs at alpha position of phosphate group. Only one nucleotide i.e. a 32P DCTP is generally radiolabeled, whereas the other nucleotides are unlabeled. E. coli DNA polymerase I is used in this method. This enzyme shows 5' 3' excitalytic activity and either removes or hydrolyzes the nucleotides from 5' side of the Nic. Nucleotides are eliminated from 5' end and simultaneously added at 3' side, resulting in movement of the Nic along the DNA. Therefore, this method is called Nic Translation. Fig. 6.14. The reaction mixture includes probe DNA to be labeled a 32P DCTP, Nic Translation buffer, the other 3 DNTPs, DNase I and E. coli DNA polymerase I. It is incubated at 140 C for 2 to 4 hours. The hybridized DNA is separated from the rest of DNTPs and used to hybridize the DNA. B. Random primed radiolabeling of probes. A.P. Feinberg and Vogelstein 1983-1984 developed this technique. The restriction fragments purified by gel electrophoresis from denatured linear or circular DNA are used to prepare probes. In this method, primers are used for DNA synthesis, but DNA nicking is not done. The purified DNA to be labeled as mixed in a buffer and heated at 100 C so that it may be denatured. Then it is put on ice to lower down the temperature. The reaction mixture containing kyano fragment of E. coli DNA POI1, 1 radiolabeled nucleotide e.g. A32P DCTP and 3 unlabeled nucleotide triphosphates is incubated at 37 C for 30 minutes to 16 hours. Since kyano DNA POL1 lacks 5' to 3' exonuclease activity, the radiolabeled probe is synthesized by primer extension but not Nic translation as described earlier. The hybridized DNA is separated from unincorporated nucleotides and used as probe for hybridization. See the figure. Figure 6.14. Nic translation of labeling 32P of DNA. See, probes developed by PCR, very small amount of template DNA can be used, therefore, this method is advantageous. In this method, the reaction mixture includes linear template DNA to be labeled, amplification buffer, Taq polymerase, primers, 1 radiolabeled and 3 unlabeled DNTPs. The hybridized DNA is isolated and used as probe. 2. Non-radioactive labeling. The non-radiolabeled DNA probes are also used in detection of desired DNA. There are 3 systems of non-radioactive routes such as using horseradish peroxidase, biotin probe and streptavidin probes as described below. A. Horseradish peroxidase HRP method. The HRP is a plant-derived enzyme which is covalently linked to DNA. Chromogenic substrates such as chloroniphthol or chemiluminescent substrates that emit light after reacting with enzyme e.g. luminol are used for detection of linked DNA. Chloronaphthol yields an insoluble purple product in the presence of peroxide and HRP. Consequently, presence of DNA is detected. Similarly, HRP oxidizes luminol and emits luminescence by using X-ray film, hence luminescence can be detected. B. Digoxigenin dig labeling system. Digoxigenin is a cardenolide steroid derived from a plant Digitaria as a hapten. The presence of digoxigenin can be detected by an antibody associated with an enzyme anti-digoxigenin alkaline phosphatase conjugate. The probe is labeled with digoxigenin 11-DUTP, which is a nucleotide triphosphate analog. This analog containing digoxigenin moiety is incorporated into DNA by Nic translation or random prime labeling. The DNA acts as probe. Anti-dig antibody to digoxigenin conjugated with alkaline phosphatase detects the dig labeled rope through enzyme-linked immunoassay. A chromogenic substrate such as 5-bromo-4-chloro-3-indolal phosphate BCIP and nitrobutane tetraethylium chloride NBT is added, which reacts with alkaline phosphatase and produce purple-blue color. Figure 6.15. A chemiluminescent substrate such as dioxetane is also used with digoxigenin or biotin-streptavidin systems. There are several derivatives of digoxigenin which emit light after enzymatic activation by alkaline phosphatase. The denatured DNA present on southern blots is hybridized by labeled probe. The membrane is placed in detection buffer containing antidigoxigenin 20. gg per milliliter, and bovine serum albumin BSA 5% with V, and incubated at 37 C for 1 hour. After incubation, membrane is washed thrice. Enzyme activity of alkaline phosphatase is detected by using BCI peak 0.17 mg per milliliter and nitrobutane tetraethylium chloride NBT 0.33 mg per milliliter as dye. Purple-blue color develops after enzymatic activation. C. Biotin-streptavidin labeling system. Biotin is vitamin H and avidin is found in egg white. Principle of this system is based on the interaction between biotin and the glycoprotein avidin. A biotin-containing nucleotide analog biotin 14-DTP or probe is prepared through Nic translation or random priming methods. Streptavidin is conjugated to alkaline phosphatase. The biotinylated probes are used to hybridize the specific sequence of DNA. The hybridized biotinylated DNA detected by specific binding of streptavidin conjugated alkaline phosphatase. Thereafter, a genic or chemiluminescent substrate is added that reacts with alkaline phosphatase and detects the hybrids. The intensity of color is proportional to the amount of biotin present in hybrid DNA. This method detects the hybrids much faster than the radioactive probes. But very small number of probes, 20 nucleotides, consist of only a small number of biotinylated sites. Therefore, there is limitation of intensity of signals. See the figure 6.15. Non-radioactive DNA labeling. K gene cloning in eukaryotes.